You roll up your sleeve, feel a quick pinch, and forget about it within the hour. Every vaccine you've ever received passed through a safety test first, and the reason you can trust that needle has almost nothing to do with modern chemistry. The real answer is far stranger. It runs on the blood of a creature that's barely changed in 400 million years, and that blood isn't even red. Horseshoe crabs bleed blue. Human blood turns red because it carries oxygen using iron in a molecule called hemoglobin. Horseshoe crab blood uses copper instead, in a molecule called hemocyanin, and copper oxidizes blue rather than red. That alone would just be a strange biological footnote. What makes it genuinely important is what's floating inside that blue blood alongside the copper. In the 1950s and 60s, the pathobiologist Frederick Bang and the medical researcher Jack Levin were working at the Marine Biological Laboratory in Woods Hole, Massachusetts, studying these animals up close. They noticed something odd. Whenever a horseshoe crab's blood came into contact with even a trace amount of bacterial contamination, it didn't just react. It clotted almost instantly, forming a solid gel around the threat within moments. The cells responsible were called amoebocytes, and the substance they produced when triggered became known as limulus amoebocyte lysate, or LAL. Why would clotting matter to anyone outside a marine biology lab? Because bacterial contamination in an injectable drug is exactly the kind of threat that can kill a person, silently, through something called endotoxin, a toxic residue left behind even after the bacteria itself is destroyed. Before Bang and Levin's discovery, testing injectable medicine for that kind of contamination meant injecting it into live rabbits and watching for a fever response, slow, expensive, and imprecise. Horseshoe crab blood offered something dramatically better. A drop of LAL reacting to a sample could reveal bacterial contamination almost immediately, with extraordinary sensitivity. By the 1970s, this test had become the industry standard, and it still is. Virtually every injectable medication, every intravenous fluid, every vaccine, and every implanted medical device manufactured anywhere gets checked using LAL before it's allowed anywhere near a human body. When COVID-19 vaccines were rushed into production and scrutinized for safety at a scale the world had never seen before, they still had to clear the same decades-old horseshoe crab test like everything else. In 2019, Jack Levin's discovery was formally recognized with a Golden Goose Award, a prize specifically created to honor obscure-seeming research that ends up saving enormous numbers of lives. So here's the twist, and it isn't a comfortable one. Meeting that demand requires catching real, living horseshoe crabs, every single year, at scale. Over 500,000 are captured annually along the Atlantic coast for this exact purpose. Roughly a third of each animal's blood is drained before it's returned to the ocean, and while most survive the process, the mortality rate is high enough, and the population pressure large enough, that horseshoe crab numbers have been declining for years. That decline doesn't stay contained to the crabs either. Migratory shorebirds like the red knot time their entire journey around feasting on horseshoe crab eggs on specific beaches. And fewer crabs means fewer eggs at exactly the moment those birds need them most. For decades, there wasn't a real alternative. Then, in 1997, the researchers Ling Ding Jeek and Bo Hao, working at the National University of Singapore, cloned the specific gene responsible for the crab's clotting reaction, creating a lab-grown version called recombinant factor C, or RFC, that could detect the same bacterial contamination without needing a single drop of actual crab blood. For years, the pharmaceutical industry was slow to adopt it, partly out of caution, partly out of inertia around a decades-old approved standard. That finally started changing very recently. In May of 2025, the United States Pharmacopeia officially approved this synthetic alternative as equally valid to the biological original, opening the door for the industry to gradually shift away from relying on wild-caught blood at all. Think about what that means the next time you get a shot and barely notice the needle. For roughly 50 years, the safety of nearly every injectable medicine on the planet has quietly depended on an animal that predates the dinosaurs, bled by hand, one crab at a time, along a shrinking number of beaches. You were never just trusting a pharmaceutical company. You were trusting an ancient blue-blooded creature that never asked to be part of your medical system, and a synthetic replacement is only now, just barely, starting to take some of that burden off its back. You roll up your sleeve, feel a quick pinch, and forget about it within the hour. And now you know what actually made that safe. Not a machine. Not a lab alone. A creature older than trees, bleeding blue, quietly guarding the door between contamination and your bloodstream this entire time.